Hi everyone, myself Dr. Kirti here. Welcome to Pathways Effort channel. Today we are going to learn about the new concept in pathology. So what if I tell you instead of giving a drug to the cancer patients, so we reprogram certain our body own immune cells to against your cancer cells. Okay, that is called as a living drug. Can you guess what it is? Yeah, it's nothing but called as a CAR therapy. So today we are going to learn about the same thing. So imagine in your body, the cancer is like a hiding thing. Okay, so who comes first? The policeman. So that is your T cells. So they come and try to recognize, but somehow these cancer cells are very smarter. They escape this mechanism. So there comes in the CAR therapy. So the scientists thought, why don't we upgrade our T cells? Okay, update or upgrade our T cells. So that is where the CAR, that is your chimeric antigen receptor T cell therapy, comes into role. So normally these T cells are like soldier in our body. They protect from immune responses, allergy, infections, and abnormal cells. So, but these cancer cells are very smarter. Somehow they break this. That is, they break the immune surveillance happens. So here they alter the antigen presentation. So they escapes from that cycle. So the cancer is not very stronger, but they are smarter enough to hide from your T cells. So why CAR T cells were created? So because of this research, the scientists created chimeric antigen receptor T cells. So they thought we will upgrade our T cells. That is like you are installing some detective app within your T cells for the easier recognition of your cancer immune cells. Okay, so that is where it comes into role. Most commonly in CD19 in B cell type of malignancies. So the normal T cells will misses your malignancies, whereas this CAR T cells will easily or earliest detection will be happen by this CAR T cell. Okay, so how do they prepare now? The difference is very simple actually when you talk. Normal T cells, they aid with the help of MHC, that is your major histocompatibility complex for detecting your cancer cells. Whereas this CAR T cells breaks that. So they don't need any helps. They are like superpower. They just directly goes and detect all these cancer cells and try to recognize and faster killing happens. So the normal T cell, where like the doctor is waiting for the lab reports. Whereas this CAR T cell is like immediately they get the lab diagnosis somehow, like LIS system or whatever it is called as. Okay, so that is a simple definition. You can differentiate between these two. So how these CAR T cells are prepared now? Let us learn in few basic steps. Okay, so the first step is collection from the patient, mainly the T cells. So how do we collect? As you know, the procedure will be leukapheresis. So you isolate all the T cells from your blood, like selecting the soldiers from the army. You are isolating all your WBCs T cells from your blood. So that is the first step. Next, what do? They insert CAR gene. What is this CAR? That is chimeric antigen receptor. Either with the help of retroviral or lentivirus vector they use, and this virus acts like a delivery vehicle. So it inserts this CAR gene into the T cell DNA. So now the T cells will express your CAR receptor, that is chimeric antigen receptor. So here we are installing some GPS tracker to detect all this cancer cells. That is a basic thing. So once this is done, then we expand this by ex vivo. Okay, so this modified cells are multiplied from few cells to millions in number, because one soldier is not enough to kill the cancer cells. We need an army. So that is what we are increasing its number. Okay, so next what do? We check the quality and the viability and how their potency, everything, the screening will be done. So then it can be frozen also, or it can be used instantly also. So how do we infuse to the patient now? So now finally these genetically engineered the CAR T cells are infused back to the patient. So now the patients will receive a living drug. So that is the one. There is no need of chemotherapy or a monoclonal antibodies needed. So this is like a customized immune system which we are giving to the patient for killing the cancer cells. So how this CAR T cells will kill the cancer now? That is the next step. Okay, so you transfer and then installed to the patient. Fine. So you genetically modifiedly engineered this CAR T cells are infused to the patient. So how they act and kill this mechanism? So inside the patient, this CAR T cells hunt the tumor cells. They go and detect all the tumor cells. So this CAR receptor will bind to CD19 antigen which is present in your tumor cells. So they kill using few mechanisms. Let us see what are those few mechanisms. Okay, so the first one is cytokine mediated release. So they release all kind of cytokine things, and they signal the immune system to kill all the cancer cells. And the second mechanism is perforin and granzyme. What do now? They made punches and holes within the cancer cells, and somehow all the cellular materials will degrade, and that is a way of destruction. And the other one is by death domain, that is your phosphate ligand pathway. As you all know, it's in the apoptosis pathway. So these are the three main mechanisms. How do they kill the cancer cells? So they recognize, they bind, they activate, and finally they kill the cancer cells. That is a whole step comes up. So what could be the side effects? As I told you, there will be cytokine release. So it can lead to cytokine storm syndrome. Release storm syndrome. So the patient will present with fever, hypotension, tachycardia, hypoxia, everything will be there. Okay, and they cause a massive cytokine storm. So immune system becomes overexcited. That is one way. They also kill your natural B cells also. So that will be a drop. And they also affect your CNS system. So it is a neurotoxicity, confusion, aphasia, and seizures, anything can come. And the last one is they usually on target of tumor toxicity. That is like the normal, as I told you, the normal B cell are also attacked here because they also have a CD19 receptor. So what will be the limitation of this CAR T cells? So the challenges will be like, antigen escape may occur, okay, it may occur, and the tumor heterogeneity. So when the heterogeneity occurs, so that failure can happen here. And the next one is like immunosuppressive microenvironment. That is also one of the reason, and the higher cost. These are all the limitations of your CAR T cells. So the cancer keeps evolving, and so the battle continues. This CAR T cell is like a personalized medicine at its peak. So we take the patient's own cell and we genetically engineer and we infuse, that will have an extra superpower, and we send them back to kill our own the patient's cancer cells. Okay, that is what happened. So not just treating a disease, but training the immune system against your own body cancer cells. That is what it happens, this CAR T cell therapy. So from patient to the lab, patient again, the same cells are infused. This is like a new mission. Okay, what are the 5 keywords I would say is, first we collect and we modify by genetic engineering, then we expand by ex vivo, then we infuse to the patient, and finally they destroy the cancer. Okay, so I hope so you understand a little bit concept about what is CAR cell therapy. So we'll see with next some other classes with new topics. Okay, until then, bye bye.